Hello, my name is Christopher Babigian, and today I'll be presenting on a recently completed pilot study focusing on the divergent motivational phenotypes across short, long, and intermittent access schedules of cocaine self-administration. As I'm sure you are aware, substance use disorder, abbreviated SUD or SUD, is a psychiatric disorder that results in severe consequences not only to the user, but on the community and healthcare system. To date, there are very few effective treatments for substance use disorder. For this reason, the Sartor Lab is interested in elucidating molecular mechanisms that play a role in addiction-related behaviors, including motivation, preferred levels of drug intake, and compulsive drug seeking. Using advanced animal models of drug addiction, we hope to uncover new potential targets for decreasing these addiction-related behaviors. In this study, we intend to determine the differences in motivation levels caused by different self-administration models of cocaine addiction, elucidate molecular mechanisms involved in compulsive drug intake and relapse, as well as identify novel and effective therapeutics for substance use disorder with a focus on epigenetic changes. For this experiment, we used adult male Sprigdolly rats that were pair housed on a reverse 12-12 light cycle with unrestricted access to food and water. Rats were implanted with a jugular catheter to allow for IV self-administration of cocaine, and then given 5 to 7 days to recover from surgery before advancing in the experiment. During acquisition training, rats were trained to press a lever in order to receive an infusion of cocaine at a dose of 0.5 milligrams per kilogram. Once behavior was learned, rats were placed into one of three schedules, either short, long, or intermittent access. Short access consists of sessions that last for 2 hours a day. Long access lasts for 6 hours a day. And lastly, intermittent access sessions last for 6 hours a day, but unlike short and long access, the animals do not have continuous access to drug. Instead, the session is divided into 30-minute bins. These bins consist of 5 minutes of drug available, followed by 25 minutes of drug unavailability. These 30-minute bins repeat for the entire duration of the 6-hour intermittent access schedule. A graphic explaining this and the resulting pharmacodynamics can be seen in Figure 1. In order to measure motivation, progressive ratio was used to determine breaking point. Breaking point is a metric that describes the highest amount of lever presses an animal is willing to pay or make in order to receive a single infusion of drug. We also conducted a more advanced behavioral economics measure called threshold. Threshold measures the rats responding as the dose administered decreases progressively throughout the schedule. Lastly, 24 hours following the final short, long, or intermittent access schedule, brain tissues were collected. The brain tissues collected include prefrontal cortex, cerebellum, dorsal striatum, and the nucleus accumbens. A complete experimental overview is shown in Figure 2. In Figure 3a, we see that during acquisition, shown as sessions -12 to 0, and before being placed into their assigned schedules, all animals had similar levels of responding. After being placed into either short, long, or intermittent access, we see a statistically significant difference in infusions between long access and both short and intermittent access schedules. In line with these findings, shown in Figure B, we see that long access rats consumed more drug than both short and intermittent access. Using the threshold schedule, we were able to obtain a metric called Q0. Q0 is a measure of the animal's preferred drug intake when cost is minimal. Here we see that intermittent access animals preferred the highest amounts of cocaine compared to long and short access animals. Next, we have breaking point. Breaking point is a metric obtained using the progressive ratio schedule. It measures the maximum amount of lever presses an animal is willing to pay or perform in order to receive a single dose of 1.5 milligrams per kilogram of cocaine. In Figure A, we see that intermittent access resulted in the highest breaking point with a statistically significant difference compared to short access animals. Using the threshold schedule, we were able to obtain a metric called Pmax, which is similar to the breaking point metric. Pmax shows the unit price at which maximum responding occurs. In Figure B, we see that intermittent access resulted in the highest Pmax compared to long and short access animals. Together, these data suggest that intermittent access to cocaine results in highest levels of motivation. Lastly, we have the alpha metric, another measurement obtained using the threshold schedule. Simply put, it is the inverse of motivation. Or more precisely, it is a measure of demand elasticity, or how quickly the demand curve falls when price is increased. In Figure 6, we see that intermittent access animals showed the lowest alpha. This suggests that intermittent access results in the highest demand elasticity, or in other words, the highest motivation. In summary, we saw that although long-access animals consumed more cocaine than both short and intermittent animals showed higher preferred consumption levels and higher motivation shown across multiple metrics. In the future, we will use the collected brain tissues to determine if epigenetic regulators are altered to a greater degree in the brains of intermittent access rats compared to both short and long access. We also intend to determine if epigenetic pharmacotherapies, such as BET inhibitors, reduce economic demand for cocaine. Lastly, we will investigate and identify cell type specific epigenetic mechanisms that drive compulsive cocaine use. With that, I'd like to thank you for listening and acknowledge the NIDA grant that made this research possible.